Welcome to Brain Ponderings, your window into the world of neuroscience research, with cutting-edge conversations with various scientists throughout the world at the forefront of research in their particular area. Today, it's my pleasure to welcome Professor Thiruma Arumugam, and he goes by Gary also. And he's a neuroscientist, professor of anatomy, physiology, and pharmacology and the Institute for Molecular Science at LaTrobe University in Sydney, Australia. He he's kind of his early research was on stroke, and then he got interested in kind of basic mechanisms of aging, Alzheimer's disease, and then because he was in my lab for a little while, he got interested in intermittent fasting and how that affects the brain and can modify vulnerability of the brain to various stresses and maybe protect against stroke or so on. So today we're going to talk about vascular dementia, so cerebral blood vessel related dementia. And this is a problem with aging mainly. It's mainly older people in their 70s, 80s in particular, sometimes earlier, but not usually. There's a few rare instances that these kinds of vascular dementia can be caused by gene mutations. Maybe we'll talk about that. And yeah, so I'm going to talk with Gary about uh, all about vascular dementia. Welcome, Gary.
Thank you. Thank you so much, Dr. Madsen. I believe this is an important topic, as you know, that NIH made it as a priority research area a few years back because vascular cognitive impairment is one of the major cause of age-related neurological problems, right?
Yeah.
And I think that it's the right move because not only in USA, around the world, with the aging population increasing over the last 20, 30 years, and it's going to increase to, quite significant. My understanding, when I reach 60 in 10 years' time, or maybe 8 years' time, and we will have almost 1.5 billion people who are aged over 60. So that's a huge population. And age-related neurological diseases such as stroke and vascular dementia affect millions of people. So it's a very important, significant problem that affects age-related, age population. Yeah, thank you.
Right. So in a stroke, like the most common kind of a stroke someone has, and that it can happen in someone fairly young too, is
Yeah.
there's a blood clot forms in a blood vessel in the brain, and then it's there. Oftentimes it resolves, so you get this, you shut off, the blood supply is shut off for some limited amount of time, and then you get reperfusion that comes back on. And most people know about this because, or symptom-wise anyway, because of the motor and sensory problems, I guess, associated with it. But stroke can also have uh, lead to cognitive problems
Yeah.
as well.
Yeah. So I think, Dr. Madsen, that one of the important thing I thought with aging, there are changes that affect the neurovascular, cerebral vasculature in the brain. That leads to a lot of problems. One of them is stroke, of course. Other one is vascular dementia, which is we are going to talk about. And I know I'm not sure how many of our audience know about the importance of or the architecture of the cerebral vasculature. It's around 400 miles long, the vessel in length. So it is quite significant amount of architecture that supply blood to the brain cells. So when we are talking now, some of our brain regions are active and they receive more blood than other regions of the brain. So the brain vasculature control blood supply to the active neurons. And then also it protects our brain. So with aging, what happened? The cells that make up brain vasculature are also affected by age-related the hallmarks such as oxidative stress, mitochondrial dysfunction, and senescence, all the age-related hallmarks you talked about for many, many years. And that lead to problem in vasculature. One of them is atherosclerosis, fatty acid deposits, and also blood clots form somewhere else towards the brain and block blood supply. That's how the ischemic stroke cause that you just mentioned. Yeah. So that's a major problem, and ischemic stroke kills millions of people every year. Second leading cause of death, if I can remember, worldwide, and kills 6-7 million people. And 1/3 of them survive, and that costs a lot of money for healthcare industries and government because of you got to take care of them for the rest of their life because they have neurological deficit. Yeah.
So if we look at the cellular architecture of a blood vessel, on the inner part of the blood vessel where the blood is flowing by, you have the endothelial cells. So it's
Yeah.
and they're connected by what are called tight junctions.
Yeah.
Right. So it's hard for it's hard for really big molecules to go from the blood
Yeah.
across to the brain tissue.
Yeah.
But so that's the vascular endothelial cells. And then you have outside of them, well, it depends where you are. In the capillaries, you don't have the smooth muscle,
No.
but
No.
the capillaries are the smallest vessels.
Yeah.
But then you get the bigger ones uh, have smooth muscle, so they can actually constrict the
Yeah.
blood vessel or
Yeah.
relax it.
Yeah.
And one of the ways that the blood vessels relax, which people listening here will probably know about, is nitric oxide.
Yeah.
Uh, there was a Nobel Prize given for the discovery that nitric oxide produced in the endothelial cells
Yeah.
diffuses to the smooth muscle cells and relaxes them, and it increases the blood flow. And so that's why if someone's having, for example, chest pain due to coronary artery disease, they're given nitroglycerin,
Yeah.
which is it's
In
explosive,
ambulance. Yeah.
but it's a nitric oxide donor. So essentially, when people take that,
It's
the
relaxes.
nitric oxide is relaxing and increasing the blood flow to
Yeah.
the heart, or also to the brain. The other aspect of that relaxation that's related to that is that drugs used for erectile dysfunction,
Yeah.
like Viagra, which is sildenafil, and others,
Mm.
they actually they don't increase nitric oxide production, but what they do is they increase the downstream events downstream of nitric oxide, namely increased levels of what's called soluble guanylate cyclase,
Yeah.
cyclic GMP,
EMP.
and
Yeah.
then that through a kinase has phosphorylation events that relaxes. Okay,
Yeah.
so um, then so with atherosclerosis, you get a buildup, you get the inflammation uh, in kind of the endothelium, the inner part, and then you get what are called foam cells, which I guess are like
Yeah.
macrophage type
Yeah. Macrophages.
things,
Yeah.
and
They
they
become sperm
accumulate the
cells. Yeah.
cholesterol, and that's what builds up.
Mm.
But then there's this vascular dementia that's called small vessel disease.
Yeah.
So, and that's what maybe we can
Yeah.
focus on here.
So just to add, Dr. Madsen, the structure of brain vessels, it's complicated. You just mentioned about, capillaries. So that the artery become arterioles and then capillary to the venules and the veins, right? So the structure is quite different between these different components of the brain vasculature. Artery, as you mentioned, have a lot more smooth muscle cells compared to veins, and arterioles also have smooth muscle cells, which highly regulated mechanism when it comes to blood supply to the active brain cells. Other than nitric oxide, I think ATP involved, which is activate P2X and P2Y receptors. and that lead to hyperpolarization and relaxation of the vessels, right, along with the nitric oxide. But and I think that noradrenaline and adenosine involved in depolarization and constriction of blood
Yeah.
vessels. So this is we call neurovascular coupling, and it's highly regulated mechanisms, and there are a lot more mechanisms, other mechanisms also involved. And while we are aging, there is the aging affect these different components of the blood vessels in a different way. That was shown by single, what do you call it, RNA sequencing, single cell RNA sequencing, the expression of genes. They looked at in normal condition as well as during the aging and age-related neurological diseases. They do express common genes and unique genes that affect or involve with their function. So it's quite interesting that that the component is different, but also they when they have, I mean, for example, endothelial cells in capillaries express different set of genes compared to artery because of the pressure. So for example, artery handle a lot more pressure compared to veins, right?
Yeah.
So they contribute to sometimes, I mean, in age-related diseases to the pathology as well. And yeah, that's one of the things. And we talked about small vascular diseases. So what really happened, these aging hallmarks of aging, right, also affect these blood vessels. And that lead to, of course, formation of small vascular diseases, but that lead to chronic cerebral hyperperfusion. That's one of the major pathological hallmarks seen in vascular dementia. As you know that, in the 1900, clinician thought the major cause of dementia due to vascular pathology, and because we didn't know about aggregation of proteins, right? And that view changed when we discovered there's a protein aggregation such as amyloid beta and tau contribute to pathology. But now we are coming back to the idea of course, vascular pathology as well as protein aggregation. So we call mixed dementia, right? That's pretty common. And in fact, a lot of biomarkers research these days trying to understand markers that indicate vascular pathology in dementia. So it's quite interesting. So the small vascular diseases causes chronic cerebral hyperperfusion, and chronic cerebral hyperperfusion drives the pathology you see in vascular dementia, such as white matter lesion, neuronal cell death, and also atrophy, brain atrophy. Yeah.
Okay, so the diagnosis then of vascular dementia is one, cognitive testing to see if there's problems with learning and memory. And my understanding is that testing similar to testing they do to diagnose Alzheimer's disease. That's one of the diagnoses. Actually, so for any of these dementias, um, it's getting better for Alzheimer's now since they can image the amyloid and
Yeah.
even the tau. But it used to be until very recently, the neurologist someone comes in and their learning and memory is clearly having big problems. It's declining. They will diagnose them with probable Alzheimer's disease or maybe probable vascular dementia based on that and some other things. But you mentioned a couple things. So you can see on MRI imaging what are called white matter hyperintensities.
Yes.
So that's the white matter is the axons bundles that are carrying information, between brain regions from one hemisphere to the other. Corpus callosum's a classic big one.
Mm.
Um, so anyway, it's just like what it says. When you look at the MRI image, there's these bright spots in the
Yeah.
white matter. And then another is kind of, I guess you'd say mini hemorrhagic strokes or microbleeds.
Microbleeds. Yeah.
And so that so the kind of the thing with these cerebrovascular dementia is it's not like in Alzheimer's, for example, the hippocampus, the entorhinal cortex are really heavily involved.
Yeah,
Uh, Other brain regions lesser. For example, the sensory or motor cortex, hardly
yeah,
any involvement. But my
yeah.
understanding with vascular dementia and particularly the small vessel disease, it can be kind of
Anyway, yeah. Mm.
all over.
That's right. So I think that age-related changes to the vasculature affect, all brain regions,
Yeah, yeah.
and therefore you can have these microbleeds. Other small vascular disease, arteriosclerosis, for example, can occur any regions of the brain, and also age-related decrease in, for example, Notch3, one of the favorite molecule we studied, right? And that regulate smooth muscle cell differentiation and so reduction in those kind of important molecules. So Notch3 can lead to reduced smooth muscle cells all over the arterioles and artery in the brain. So and that's how some of the pathology lead to not only microbleeds, but also other small vascular diseases that cause to reduce blood supply. So you are right, like unlike Alzheimer's disease, vascular dementia can affect different brain regions, not very specific to the hippocampus. But what we know from um, the brain bank in UK, Dr. Madsen, that when I had a chat with a clinician recently at Monash University, and he mentioned that when they looked into the brains of vascular dementia patients, these are postmortem tissue, there's a hippocampal atrophy you see.
Hmm.
So the hippocampus probably also, affected in vascular dementia uh, earlier on compared to other brain region, which is we need to do a lot more research and look into the
Maybe
postmortem
that
tissue.
makes sense because as you know, and maybe we'll soon talk about the animal models, right, where you can
Yeah,
really get at what's
yeah,
going on. As and right. So sometimes people, for example, they have heart attack, cardiac arrest, and the
yeah,
blood supply is shut off to their brain, the entire brain, or not completely necessarily, but it's reduced for even a couple minutes,
yeah,
right? And they may have some permanent brain damage and corresponding uh, deficits,
yeah,
which can be cognitive. And
yeah,
so in that, there's an animal model of whole brain hypoperfusion,
yeah,
right? You clamp off both carotid arteries for a while.
yeah,
And what happens there is hippocampus is pretty vulnerable.
yeah,
Yeah.
yeah. This goes back to your finding early days. I mean, if I can remember that you mentioned or you published an article where hippocampal neurons are more active, is that right? Because they are involved memory formation all the time. Is that right? Like they are more active than other brain cells. Therefore, they may
Uh,
be.
yeah, I would say that. And as globally, and then also the interneurons are particularly active. Uh, the inhibitory GABAergic neurons that keep
Yeah,
prevent hyperexcitability. And we think in stroke, maybe in vascular dementia, there's too much hyperexcitability and excitotoxic death occurring.
yeah,
Uh,
yeah. In the hippocampus, and that's possible. And this so the animal models what we have, we can talk about that one. It's also reduced blood supply to the whole brain. And we look into cortex and the hippocampus just to compare with the human pathology. What we see, we don't see much cell death or reduce cell count, neuronal cell count in the cortex, but we do see in the hippocampus. So the hippocampus probably the first set of neurons affected in response to chronic cerebral hyperperfusion in animal models too. Yeah,
yeah.
yeah,
Okay. Yep. Yep.
yeah,
Right. I did want to mention, you mentioned this notch three. So this is I've had some interest. There's this family of genes and the proteins encoded by the genes called the notches.
Yeah,
I think the one that's been studied most is notch one, which is
that's
very
one. Yeah,
important in development.
yeah.
And it turns out that that one of the enzymes that cleaves the amyloid precursor protein
Mm.
to generate the amyloid beta peptide uh,
It's
also
also clear
cleaves
much.
Notch. Notch is a
Yeah,
transmembrane protein. It sits in the outer membrane of the cell,
yeah,
and then normally during development, Notch one is there's certain signals that activate it. There's ligands called jagged or so on, and it plays important roles in controlling cell proliferation, differentiation, migration. And
yeah.
so this Notch 3 is related to Notch 1. And this disorder is called, I don't remember the full name,
Cadasil, Cadasil.
CADASIL. It's CADASIL.
Yeah, yeah.
But you know what each letter stands for? I can't remember. Cerebral.
Yeah,
Am- am- am-
sorry, I'm same here.
yeah, yeah.
Uh, our friend Tang should know because he's focusing on CADASIL for a long time, and he has group of patients with the family history of CADASIL, and yeah, the
It's mainly, it
CADASIL is
was
the not.
discovered in, I think, Japanese family or some somewhere in East Asia. I'm pretty sure about that.
Yeah. Oh, yeah. There are a lot of families in Taiwan for sure. And yeah, I can't remember where they discovered, but Notch 3 specifically expressed on, when it comes to brain vasculature, smooth muscle cells, where Notch 1, in endothelial cells as well as in neurons. We were focusing on neurons,
Yeah.
right, when we looked into the role of Notch 1 in stroke. And we haven't looked into role of Notch 1 in vascular dementia.
And so,
But
so these people with Notch 3 mutation, they die at a fairly early age, I would assume,
yeah,
and they get did,
the
did they
microbleeds and
yeah
microbleeds all over the brain. Yeah, that's because it's similar to what happened with the aging, right? If the Notch 3 expression goes down, that leads to reduction in smooth muscle cell differentiation, and that affect the arterioles and arteries, and that's a similar thing what happened in the Notch 3 mutation patients there. They have microbleeds because of there's not, it's affect the smooth muscle cells differentiation.
so it's a
Uh,
loss of function mutation,
yeah, loss of function mutation. Yeah.
right? And so then did the person have to be inherit a mutant Notch 3 from both parents or just one? Is having 50% reduction in normal Notch 3 sufficient to
Yeah,
cause?
that part I'm not sure, Dr. Madsen, but I
Okay.
know patients survive for a long time compared to some patients survive for a long time compared to other patients,
Ah,
but
ah.
that's true. And also, Tang and myself wanted to initiate biomarker study differentiating these patients who are showing pathology earlier on compared to some patients, so the CADASIL pathology a lot
So
in
so
later
so
part of
just
their age.
for so
Yeah.
Tang he was a postdoc in my lab from Taiwan, and he was going to do it. I haven't communicated with him in years. He was going to do a trial of intermittent fasting in
Yeah.
stroke patients, but that I don't think that ever happened.
No, he registered for this trial and wanted to initiate. I think there are a few patients. My understanding he started, but never went on the way he
Uh,
wanted. So I'll get back to him and see where he is at
Yeah,
with the trial. Yeah,
tell him
yeah,
I'll be a cheerleader for it. But maybe vascular dementia is another one that might be worth.
yeah. I mean, we published couple of papers in the last few years how intermittent fasting can protect against vascular dementia, right? We can talk about that
I mean,
later on. Um,
let me quickly mention a couple
yeah,
other things at the genetics. So,
yeah,
ApoE4, which is a risk factor for Alzheimer's disease.
yeah.
It's also risk factor for vascular dementia.
Um,
I think so.
ApoE can be risk factor, risk factor for vascular dementia. But in that area, I haven't come across many papers that associate vascular dementia patient with ApoE
Ah,
mutation,
hmm.
which I probably looked into. I'm not sure, Dr. Madsen, that, but if you think about that, how ApoE can cause the pathology, it's possible, right?
Yeah, and also in some cases of vascular dementia, there's amyloid accumulation uh, in blood vessels,
Yeah, yeah,
right?
yeah, that caused a problem. Yeah, because this is part of one theory how early deposit along the blood vessel can interfere with function of the blood vessel, and that can also lead to some of the small vascular diseases, right?
Yeah,
And which is well reviewed in a recent paper published in Neurons, sorry, and that's possible. And when it comes back to amyloid beta aggregation, but there are the models showing that probably the dysfunction of blood vessels before amyloid beta aggregation. So, and this is the area also under research now when it comes to biomarker studies, as I mentioned before. So there are a few things that need to be cleared in the literature by doing basic research. And one of them, the link between the vascular dysfunction and amyloid beta aggregation, which we know based on clinical sample, there are correlation, and most of the patients are from both conditions. Yeah,
yeah.
yeah.
All right, and then so the major risk factors, well, age we've mentioned. What are the other risk factors for vascular dementia?
Oh, hypertension, diabetes, for example,
Yeah,
age-related metabolic disorders, right? Yeah.
yeah,
And the hypertension increased blood supply to the brain earlier on, but later that leads to vascular dysfunction.
yeah.
It can affect the microvessels in the brain, and so that lead to hypoperfusion, right?
Yeah.
And hypoperfusion is the major hallmark, as I mentioned, for vascular dementia. Yeah.
So hypertension uh, promotes damage essentially to the blood vessels, and then that leads to
Yeah. Hypoperfusion.
hypoperfusion.
Yeah.
Okay, and
And
then
yeah. So
diabetes, insulin resistance, um, high cholesterol levels, I guess,
yeah, high cholesterol
hypercholesterolemia.
levels can cause arteriosclerosis, as I mentioned. Um, that one of the major cause of hyperperfusion, and
There's
also
also arteriosclerosis,
arterio. Yeah, sorry, arterio. Yeah,
which
yeah,
is used to be called hardening of the arteries.
yeah, hardening of the artery. Yeah,
But is
and
that you get calcium deposits um, and also there's the proteins that they're called extracellular matrix proteins. There's something called fibrosis.
yeah, fibrosis also can affect.
It's almost like scar tissue,
Yeah, yeah, that's
collagen, yeah,
deposit. Yeah,
extracellular matrix
matrix. Yeah, that
accumulate.
also to small vascular diseases. Yeah.
There's
So there.
one other
Yeah.
thing is atrial fibrillation. I saw somewhere where that's a risk factor if you have atrial fibrillation. That kind of makes sense.
we because of irregular
Irregular
blood
heartbeat.
flow,
Yeah.
yeah, heartbeat, and yeah, how that directly cause. I'm
Well,
just thinking.
if it's chronic, it would make, if you repeatedly have that. I don't
It
know.
can.
I was reading up on it a little today.
Yeah, it probably can affect the blood flow regulation to the brain area, and then may lead to hypoperfusion, right? That's possible. Yeah.
Yeah. All right. So, and then obesity obviously is a risk
Yeah.
factor. Okay. And then how to reduce one's risk? Again, this is age related. So, and like with Alzheimer's disease, undoubtedly, by the time someone goes to the clinic and gets diagnosed, there's already a lot of damage done. So ideally, uh, want to we want to start early
Yeah.
in terms of what can be done. And there are things that can be done, people can do in their everyday lives. And they're all related to the risk factors you mentioned. You want to talk about the different interventions?
Yes. So one thing we know, Dr. Matson, that with aging, right, we do have reduced cerebral blood flow to the brain, right? And it's my understanding um, around 5% every decades decrease in blood flow, that almost 5% reduction in oxygen and glucose to the brain tissue. So we have a lot of evidence from animal studies and human data that blood flow reduces. And in fact, we published a paper a couple of years back in Aging, where we looked into the blood flow to the brain using laser Doppler, right? And you can see clearly huge difference between young animals and aged animals. And the similarity is, so when you put these coils around the common carotid artery to reduce the blood supply, that's the model for vascular dementia, and that reduced the blood supply by 30-40%. So the aged animals' blood flow similar to young animal with a
Oh,
coil. Yeah, that's quite interesting. So this true for human as well. We have reduced every decade around 5% reduction to the blood supply to the brain, and that lead to problem with the neurovascular coupling and cause synaptic loss. Just want to mention about synaptic loss, and in this model, what I talked about right now, which is the coil around the common carotid artery, reduce the blood supply to the brain by 30%. And we looked into the effect of, I'll come back to this one, intermittent fasting. First, we looked at compared to the normal control sham animals to the hypoperfusion animals. We found that synaptic loss in the hippocampus, and that was reversed by intermittent fasting. So along that note, so intermittent fasting, calorie restriction, exercise, these are our favorite topics of discussion all the time.
and
Yeah.
then Gary, in that experiment, you maintain the animals on intermittent fasting before you uh, reduce the blood side of the brain?
Yeah.
Have you
Yeah.
tried to see if you can enhance
Reverse.
recovery?
Yeah. So all our experiments so far using this model, vascular dementia model, basically we put the animal for a few months, 3 to 6 months, that different set of experiment have different time frame. So this particular around 4 months intermittent fasting, then we induce the vascular dementia. So bilateral common carotid artery stenosis, right, by putting the
Yep.
coils. And then we maintain throughout the 1 month period with the coil. So, but we haven't done the post because intermittent fasting yet. That's quite.
And have you done it in let the animals get old or fairly old at a point where they should be having some problems? Uh, yeah, and then do the intermittent fasting
Yeah.
for like whatever 4 6 months, and then compare. Don't even do the.
No. No, that's I really want to do that. In fact, we have two sets of experiment at the moment. I'm pretty sure you like this one. We put this animal for throughout their lifespan under intermittent fasting, right? Like so up to 2 years. Now we finished 18 months tissue collection. We are looking at a lot of different things, doing all the sequencing, like from WGBS, RNA sequencing, and proteomics to understand the remodeling of the brain, not really vasculature, but the whole brain itself. And also we are doing electron microscopy to understand about the changes in the brain, structural changes to the neurons and mitochondria and other cell types. So we are trying to understand what's happening throughout the aging to the brain. So we have different time points, and also we are trying to see how intermittent fasting remodel the brain to protect against age-related, decline, the structural and functional decline. So we are also doing cognitive impairment test, Barnes maze, for example. So this is going to be quite interesting because we do have different time points throughout the aging and also the whole molecular, structural, anatomical changes. So you'd like this paper to come, Yeah, yeah,
Sounds good. And okay, so intermittent fasting, physical exercise.
yeah. Calorie restriction and phytochemicals, phytochemicals, your favorite topic, and we talked about, and there are phytochemicals that work against some of the hallmarks that drive
So maybe
aging.
plant-based diet is I guess if you adjust for age, is vascular dementia less common in areas where, well, I guess it would be like the blue zones or areas where people have mainly a plant-based diet and live at the time. So
Yeah,
is there data on epidemiological studies looking at prevalence of vascular dementia and diet?
I'm not sure, Dr. Madsen, specifically for vascular dementia, but we know for sure, for example, Okinawan population, right? Their cognitive uh, impairment much lower compared to the other part of the world, and then they do practice kind of calorie restriction diet, and also it goes back to the Italian group where they practice intermittent fasting type of, where they don't eat after sunset and then the first meal around 10 a.m. They also suffer less from age-related diseases, including age-related neurological diseases. So,
Is that
um,
Sardinia or somewhere else in
yeah,
Sardinia?
that's right. Yeah,
Yeah.
and there's a Mormon group in USA, and then Korean monks, and there are a few groups who practices either calorie restriction or intermittent fasting type of diet, and
Oh,
yeah,
also another risk factor I saw is drinking a lot of alcohol.
yeah. That's known because that cause so the brain images, I think, is that MRI or PET scan show that a lot more atrophy in the brain compared to normal, so it's quite well established that chronic alcohol intake cause probably excessive amount, not one or two wine per day, I guess.
Yeah, more
um,
than that. Yeah,
More than that, yeah, yeah,
yeah.
yeah,
All right,
and yeah.
go ahead.
I was thinking that uh, when it comes to intermittent fasting and vascular dementia, um, we have shown that intermittent fasting protects against white matter lesion and neuronal loss, and also blood-brain barrier damage, where we found that the tight junctional protein are intact when you put animal under intermittent fasting compared to the control. So the mechanism is pleiotropic, not towards one type of cells, as you see in other disease conditions. So and that's why I think that it's important to start the lifestyle modification that can help against age-related neurological diseases, instead of we wait until we get this disease and go to hospital and try to find, that's not going to work, right?
No, no.
Yeah,
And right, another risk factor was this is something we were interested in a long time ago, and we did some studies on it, and then we kind of quit because we were pretty convinced it's true, and that is high homocysteine levels.
yeah, yeah. I
Um,
read you published some of the paper, um, in 90s, right? High level of homocysteine level
Yeah,
and stroke. So
we
stroke,
were within a couple year period. There was the famous Framingham study up in Boston. It's a
mm,
study, epidemiological study that linked high cholesterol to increased heart attack risk, and they showed that people with high homocysteine levels are at increased risk for
mm,
Alzheimer's disease. And then we did some simple studies. We exposed neurons to different concentrations of homocysteine alone, or in combination with amyloid beta peptide, or in combination with glutamate, excitatory amino acid that can
mm,
damage cells if they're excited too much. And bottom line is we found that homocysteine can make neurons more vulnerable to being damaged by amyloid and glutamate. Uh,
mm.
and homocysteine levels can be lowered by taking folic acid, folate,
Yeah.
in the diet. Now, most Americans and I guess most modern countries they actually put folate in bread and I don't know other things. It's certainly in all multivitamins.
Yeah.
But if you do get folic acid deficiency, that's not good. So that's one. I don't necessarily recommend
Mm.
dietary supplements, except a multivitamin's good, and folate is a good thing to take, I think.
Yeah. So just homocysteine promote the neuronal cell, death or it's interfere. I mean, I'm just trying to understand. I forgot, Dr. Madsen, the mechanism exactly. They cause damage to the membrane. Is that
Well,
right? Or
it
yeah,
it, it promotes oxidative stress, essentially. It's
yeah.
involved in one carbon metabolism
Okay, yeah,
and uh, methionine
yeah.
metabolism, and
Okay.
so actually, and I had Dudley Lamming on from University of Wisconsin recently to talk about mTOR and
Mm,
protein and amino acid intake and these branched chain amino acids, and then some other one. It's been shown pretty and reproducibly in at least in mice that uh, if you reduce methionine, just that one amino acid in the diet, the animals live longer.
mm,
So protein restriction, methionine, and
yeah,
particularly certain amino acids, and
yeah,
that could be part of the way that so folate's involved in one carbon metabolism, and homocysteine is a product. The other thing is there's these certain genes and code proteins involved in this cycle
yeah.
of homocysteine production from methionine, and if you knock those genes out in mice, it's not good for their brains.
Yeah.
Yeah.
And along the line, Dr. Madsen, that you worked on NAD plus, right? NAD plus also reduce with age, right? And that important molecules involved in epigenetic sirtuins activity, right? And then
Yeah.
and sirtuin 1, 3, and 6, I guess.
Yeah.
And then that's important mechanism that regulate deacetylation from histones. And so I was thinking over the years, like coming back to intermittent fasting research, but also when we age, the way it's affect the vasculature or neurovasculome, probably driven by the changes in metabolites,
Yeah.
and that changes in metabolites, and some of them are, important that promote epigenetic mechanisms
Hmm.
and change the epigenetic landscape and drive chromosome accessibility to express genes, like and then of course protein. And this is if it's true for any environmental changes that affect our body and cells or cells in our body that act through the metabolites, epigenetic cascade, gene expression, and then protein. It's also true when it comes to this lifestyle modification, and that's a big question mark in the literature. How, for example, intermittent fasting, exercise, color restriction, and other good practices that help against neurological diseases or age-related diseases may function. So may act to protect, right? So this is one of my interests in the future to look into the mechanisms. So that's why we are doing a lot of sequencing, including metabolimics, to understand the overall mechanism, how everything initiated, not only by practicing good lifestyle, like color restriction and intermittent fasting, but also during aging. It's probably opposite, other way around. So trying to understand what are the metabolites during aging, increasing or decreasing, and how intermittent fasting and color restriction can bring it back.
Yeah, well,
So yeah.
that's something I've written about quite extensively, most recently in a Nature metabolism paper. Uh, I think the title was the uh, intermittent metabolic switching theory of intermittent fasting. But
Mm,
the ideas are in another paper. It's kind of the same thing that catabolic anabolic cycling model. But it seems like everything that protects the brain, the heart, any all our organ systems, kind of counteracts aging. They all involve transient imposition of a stress, a good
mm,
stress, EU
yeah,
stress on cells, and then a recovery period. So you have challenge recovery. So exercise, rest,
yeah,
fast, eat, and sleep is recovery for all of those. Or, you mentioned these phytochemicals, chemicals in vegetables, fruits, mushrooms, uh, so plant and fungi-based
yeah,
diets. These
yeah,
these chemicals are in those plants or fungi because their function is to keep insects,
yeah,
deer, us from eating
yeah,
much of them, particularly the vital parts. So if you and maybe I should have a podcast on this. I could talk forever on this, as you know. But
yeah, yeah,
um, right. So a couple examples. Sulforaphane,
yeah,
high concentration in broccoli sprouts, some
yeah,
other sprouts. Quercetin, high
yeah,
concentration in the skin
mm,
of apples, for example.
yeah,
Um, Curcumin, high concentration in root turmeric
yeah, yeah,
root. Caffeine, high
yeah,
concentration in coffee and tea.
yeah,
So if you take any of those things, if you take broccoli sprouts, turmeric root, tea leaves, ground up coffee beans, and you put
mm,
them on your kitchen table
yeah,
and say you have ants in your house, the ants won't touch them.
no, yeah,
If you put anything with sugar,
they,
ants will go crazy.
yes,
We did a nice study, and I don't know, it was a small part of this actually, but there's a chemical called tomatidine, which is it's present at high concentrations in the skin of green tomatoes.
wow, mm,
And then as the tomatoes ripen and turn red, its concentration decreases.
yeah,
Okay.
yeah,
So what this chemical is doing is in the green tomatoes, it's preventing insects from, or birds
ah,
or whatever from eating the tomato. And
mm,
then its levels go down. And then because in the green tomato, the seeds are not mature.
no, no,
And right, but once the fruit
yeah,
ripens, doesn't matter what fruit, then the
yeah,
seeds are mature and you can grow something and they can be spread.
yeah,
So,
reproduction,
so evolution, these
Amazing.
adaptations are quite amazing. But anyway, the point is that all these things that seem to be good for brain and body health, they involve exposing yourself and your system
Yeah,
to stresses, whether it's metabolic through fasting or exercise, or chemical with these natural chemicals we evolved consuming.
yeah,
Um, yeah, and then
yeah,
so then those induce adaptive stress response and recovery. So during the stress period, challenge period, cells
mm.
upregulate autophagy. They upregulate antioxidant defenses
And
and
trophy
so on.
factors.
And so they clear out the damaged molecules and organelles. And then during the recovery period, cells can grow, new synapses can form, new
Yeah,
uh, mitochondrial biogenesis. But if you don't have the challenge period, then
you don't produce.
none
Yeah.
of the good things happen during the recovery period.
Yeah, this is evident from our sequencing studies. So we have done a lot of sequencing over the years, right? And also proteomics and phosphoproteomics. And we do see, expression of genes and proteins that support everything you just mentioned, Dr. Madsen. So you see that the good protein, the good genes that are upregulated in response to intermittent fasting in our case, and it's also true for color restriction, I guess. But there are a lot more studies to be published in the near future. Of course, you are part of a couple of other studies. I was sending the papers to you, right? So we are identifying new mechanisms how these challenges can lead to expression of much more than what we know in the literature. One of the things that we found recently in the liver, serpin 1A, right, which
Ah,
is very important protein that involved in liver function. So if the deficiency of that protein cause liver dysfunction and liver diseases, what we see intermittent fasting increase in liver, the level of serpin 1A, where in the muscles and the brain, other way around. Then if you look at the literature, that it's good when it comes to liver function, but maybe not good for other tissues, and which is still need to be, well researched to understand the mechanism of this protein in the brain and muscles. So the way the lifestyle modification works, not only pleiotropic, but also kind of organ specific. It's amazing. That's it's coming back. I don't know, maybe coming back to evolution, how we evolved for millions of
ah.
years.
And then the communication between the organ systems. For
Yeah.
example, if you impose stress on, the ischemic preconditioning. So
Mm.
there's this called remote conditioning,
Yep.
where if I were to like put a blood pressure cuff on my arm and tighten it
Yep.
up so it shuts off the blood supply for like 5 or 10 minutes, so your arm starts to hurt because of ischemia.
Yes.
And then there's been studies showing there's animal models for this, that if one induces stress uh,
Yeah.
in one area of the body, you can get adaptive beneficial
To the
effects
brain.
in other parts or even the brain.
Yeah.
So
In
that,
fact, Dr. Madsen, David Hess at Augusta University, he's the dean of Medical College of Georgia. His group doing this fund for a long time, and my understanding they are also applying this in patients,
yeah,
stroke patients. Yeah. So it's a preconditioning homeostasis, right?
yeah,
Yeah. Yeah. So
yeah.
yeah, that lead to expression of genes that prepare your
Well,
cells.
probably there's two theories. The most obvious one is that there are proteins or other molecules released from the stressed arm tissue
Oh.
into the blood. Then they go to the brain and affect in a good ways the vascular cells, the neurons, or whatever.
Mm,
We actually know this for a few things like vascular endothelial growth factor. I think one
yeah.
thing that have your brain, the blood vessels in your brain, just like in your muscles,
Yeah.
they can grow, and they can be stimulated to grow by stress, by exercising the muscle or exercising your brain cells.
Yeah.
With aging, that we call it angiogenesis ability declines,
Yeah.
but it can be stimulated by exercise. I don't know if
This is
anyone's looked with intermittent fasting actually at angiogenesis in the brain.
Oh yeah, I mean we have data I can share with you. We looked into the effect of intermittent fasting in brain vasculature and did RNA sequencing. I haven't published it yet. Some amazing set of genes upregulated in the blood vessels of the brain, right? But coming back to your BEGF,
Right,
vascular,
but have you actually quantified structurally angiogenesis, the density of the microvessels? There's good
um,
antibodies you can stain
yeah.
the blood vessel.
Yeah, we have done, in fact, in response to stroke a long time back with Justin Lathia. But we haven't done in response to intermittent fasting. That's a good one. We should do because we have tissues at the moment
Yeah,
from aging mice, and we can put collagen 4, right? For example, is
yeah,
that collagen 4? Yeah.
yeah.
The Dr. Madsen, you just mentioned BEGF, right? Vascular endothelial growth factor. So my understanding, based on the literature, that the decoy for the receptor decoy increase with aging, which interfere with the VEGF
Ah,
signaling, right? And
yeah.
that also affect the vasculature um, during aging. But there's a study treating with the VEGF increase the lifespan by improving the brain vasculature. That was also published, right? Yeah, it's quite
Oh, I didn't
interesting.
see that.
Yeah,
No,
as I came
no.
across some time back. Yeah, and that not restructure the blood, sorry, the brain vasculature and also increase the cerebral blood flow, if I can remember correctly. So the VEGF and angiogenesis goes down with aging because of the decoy in the blood, and it brings back our young and old plasma, right? That the experiments, parabiosis, right? When they put
Yeah,
young plasma, I'm not sure they have looked into the brain vasculature in those animals. Yeah,
I haven't seen it.
yeah,
I had Tony Wascori on my
yeah.
podcast probably, I don't know, year and a half, 2 years ago, and they did a small trial in Alzheimer's patients of, transfusing young blood
Mm,
uh, into them. And my recollection, they didn't see any obvious. But again, by that time, there's already a lot of damage. I'm still skeptical that we're going to get certainly not going to get any recovery to go back the way they were before they had
yeah,
cognitive impairment. But even slowing the rate of decline is not easy.
no.
Because
I think this is
most people with Alzheimer's are in their 70s and 80s.
80s. Yeah, that's the damage is already done for a
Yeah,
couple of decades or more, right? So
yeah,
this were the Jack model. When they talk about vascular damage, then amyloid beta and tau, and then cognitive impairment. So by the time we go to the hospital, it's too late.
yeah.
Uh, yeah, yeah,
So
yeah,
for people with high blood pressure, and there's genetic factors too for high blood pressure,
mm,
diabetes, somewhat for obesity, although not as much as you might
yeah,
think. So if you have high blood pressure and you aren't controlling it good with exercise,
yeah.
intermittent fasting, you should take. There are good
High
drugs
blood
for
pressure
keeping
medication. Yeah,
blood pressure in normal range. Then diabetes, again,
metformin.
most people, if you catch it early, you can get rid of it with exercise and calorie restriction and intermittent fasting. Uh,
In fact, there's a human trial, right? Human studies where the pre-diabetes for 5 weeks or 6 weeks on 2 p.m. to 8 a.m. intermittent fasting, time
mhm.
restricted feeding, and they got all the markers back to
Yep.
normal.
That's
And
just with daily time restricted eating.
Yeah.
And if you combine it, if you add exercise, you even get better.
Yeah.
Yeah.
And one thing I would like to mention since you talked about diabetes, the metformin, right? Metformin is one of the
Yeah.
most used drug against diabetes. And there is a study published recently in monkeys, I guess, and published in Cell, I guess, showed that the metformin um, basically protects against brain aging by significant time frame. Have you come across that study?
Do you know where it was from? What?
From China.
No.
Um, yeah, it's a big study.
I got to tell you, Gary,
Yeah,
since I retired in 2019, yeah, almost 6 years ago,
yeah,
if you go on PubMed now, and it doesn't seem to matter what I put in for keyword search terms,
yeah,
there's been just an explosion of papers coming out of China.
yeah,
And, you know, I, when that happens, I just wonder what are they
yeah.
really doing that much high quality research, or what's going on with that? But anyway,
Yeah. I mean, there are two factors, Dr. Madsen, because I traveled to China recently. Amount of funding increased significantly beyond our imagination. So there are a lot of good researchers are doing research, but there are also paper mills, right? So
yeah,
Uh, yeah, there are a couple of issues, but I think that the Chinese government trying to control the paper mills
right,
recently published in Nature, and but there are a lot of good researchers
yeah.
doing excellent research these
And
days.
by the way, there's Americans moving to China,
Yeah, yeah,
and and a lot of people who came from China here to get a PhD and do postdoc and get a faculty position here, they're going back to China.
going back. Yeah, they're
And
going
here
back
and
and
here
then
and here this country is
cutting.
doing cutting research. It's a
It's
big
bad.
mistake.
Yeah,
Big
I
mistake.
mean, I talked about recently when I went to Birmingham, Alabama, right, and um, at this time point of history. It's not smart to cut funding in USA because you are competing with China, right? And where amount of funding is significantly increased, and then
Yep.
what's happening in USA, it's not very healthy in years to come, I guess.
You know,
I guess you would
I
agree.
said, but we're making a mistake. Actually, I have my doubts about whether it is a mistake. I think it's intentional.
Hmm.
We are, this current administration is fascist. They, so first uh, Bertrand Russell, he's a famous philosopher from the UK, and, and a historian too.
Yeah,
And he had some comment like, with regard to the fascist playbook. First, he used the word fascinate and make fearful the general public and get them to
yeah.
vote for them. Then once
Hmm.
they get in office, they they muffle, you know that word muffle the intelligent people.
Uh-huh.
And that's what's happening here. The universities are being muffled. And
Yeah,
anyone who speaks out, speaks
yeah.
truth to power, they're squelching. That's
So why?
this is happening. And hopefully most people in this country are beginning to recognize it, but maybe not
Yeah.
because they didn't recognize it when they voted in the last election, and it was clear,
Mm.
since 2016 this was going to happen. So anyway,
It's
let's get off the politics and get back to.
yes, yes, yes.
But it's
I
actually,
think.
yeah, it's a big problem. I think anytime you you suppress uh, people's questioning and
No, it's not good.
discovery and trying to find out the truth about things, in big
Yeah.
trouble. All right. So then there was another thing, right? So we high blood pressure, diabetes. You mentioned metformin, the GLP-1
Yeah,
receptor
small molecule
agonists.
rapamycin, another one. Yeah.
They seem to
Um,
act mainly by suppressing appetite. So my own opinion is that their main effect is by caloric restriction there.
restriction.
Yeah,
That's
yeah, they're
that's what I
suppressing
thought about.
appetite so the people aren't eating and they're losing weight. That's
Yeah, yeah,
how else are you going to lose weight?
yeah. No
Well,
calorie.
you can exercise, but these people with obesity, for the most part, the ones that are taking GLP-1 agonists, they're
Yeah,
pretty sedentary people. So, and they're losing a lot of weight, and it's because their appetite, it's just
reduce eating less,
goes
calorie
down.
restriction.
So
Mm.
it comes back to the same thing, a drug now suppressing appetite, and I don't know. We'll see how in the long term how safe they are. I don't like the idea of people just
Yeah.
taking a drug rather than putting effort into not eating so much and exercising. But
I found I have few friends or family members who are taking it's really hard for them to get back to the good lifestyle we talk about, so they have to rely on these drugs. That's the only way. And but I agree with you.
well, I,
Yeah,
there has to be a mechanism. So what I would have done, like if I was in charge of the government, there would be a couple of things. So the healthcare would be focused on prevention, risk reduction, right? And so there'd be a lot more primary care physicians. And then in theory, in the long run, we need fewer specialists, right? And then
yeah.
they would prescribe
Mm.
specific eating patterns, specific diet
Yeah,
composition, specific exercise routine, maybe graded thing over a period of months.
yeah,
They would follow up, or their office assistants follow up with the patients,
yeah.
early on like a bi-weekly, weekly basis, and
Mm.
then monthly basis. Then for people who can't adopt these new lifestyle
Mm.
habits, I guess you'd say,
Yeah,
then there'd be government paid, kind of like rehab. 1 month, they go into a place for 1 month,
yeah.
where there are, experts on exercise, intermittent fasting, diet composition. And there's other people there too, the same situation. They have obesity, right? They can't change their habits. And my guess, based on the clinical trials I've been involved with
Yeah,
and talking to other clinicians and so on, is that I don't know what percentage, but a good percentage, maybe
yeah.
half of the people at the end of that month, they will have switched. Now, the other
Mm.
thing, the problem is then they go back to their home environment
Yeah,
where their spouse or whoever is eating junk food, not exercising. So there has to be a focus on education too.
yeah.
And the parents and the family members, it's kind of like, you know, drug rehab or something like that, or someone with severe depression that if you can get the family involved and
Yeah.
then yeah, so I had a doctor up in New York who works with children with obesity. So these are
Yeah.
kids like 10 years old, you know,
Wow.
10, 12, 14 years old with obesity. And he had
Mm.
some success in he'd get the parents and the parents and the kid, and he'd get all of them to switch their eating pattern to daily time restricted eating.
Mm.
And he's having success because the kids, when the kid's home,
Yeah.
the parents are eating the same
Okay.
routine,
Yeah.
eating
That helps.
pattern,
Yeah.
right?
Yeah.
And I think it's the same true with exercise. If your parents exercise, you're more likely to exercise.
Exercise. Yeah.
Yeah.
I agree. And this is also important when it comes to the next generation, right? Because that's a paper we just published in International Journal of Biological Sciences. Dr. Madsen, you are co-author, of course, where we showed that the parental intermittent fasting affects children's
Yeah.
metabolic health. And that's directly also probably involved with the neurovascular aging and associated neurological diseases. This is what we want to do in the future as well, look into how well the children from intermittent fasting parents perform when it comes to neurological diseases. And what we are doing at the moment, with the help of the Jim Penman, who is you talked to a while ago, and we just started transgenerational 10 generation intermittent fasting and calorie restriction study.
10
So
generations.
10 generation, that's the ethics approval we got, and we want to see up to 4 generation and see how that goes. And then, if we get good results up to 4 generation, we'll continue up to 10 generation.
And of course, in the animals, you're not looking at the environment, you're looking at epigenetics, that is,
Yeah.
changes in the, yeah,
DNA methylation. So
yeah,
that's
the genome of the mother or father that are
yeah,
uh, then transferred to the kids. So they have a
yeah,
pattern of gene expression that's different than if the mother or father would have been healthier.
yeah.
Yeah.
We have already some data.
Yeah.
This is an intergenerational study where we looked into the DNA methylation pattern in response to intermittent fasting in both parents, mother and father, and from both group, intermittent fasting or ad libitum, and looked at offspring from these animals, and we can see there are significantly different, DNA methylation
Yeah.
pattern between parent animal as well as offspring. And we also collected sperm and tried to understand if that, you see similar pattern of DNA methylation in sperm, and you can correlate the parental tissue, sperm, and offspring tissue. So it's very exciting time in the lab. We are trying to understand the mechanism of intergenerational inheritance, possibly transgenerational in the future. But all these changes, how that
This
affect.
is very important because
Yeah.
it's very clear in humans. So it- it's very clear with women, and there's some evidence for men too, that if the woman has obesity or diabetes during
Yeah.
pregnancy, their child has poor health outcomes,
Mm.
kind of globally poor health outcomes. There's even some evidence that
Yeah.
it's an autism risk.
Yeah.
And then there are animal studies suggesting that the metabolic health of the father can also affect, through, and these are, so what's happening is apparently, so it's either in the egg or the sperm, or maybe possibly even in the early, yeah, it's probably in the egg or the sperm. So the environment of the egg and the ovaries of a female with obesity, diabetes is not good. There may be increased
Mm.
inflammation or metabolic insulin resistance, whatever, that lead to these changes in the chromatin and the
Mm.
ways in which genes are expressed, and
Yeah.
then they can be methylation, they can be passed on.
Yeah.
Yeah,
There's a big knowledge gap how this information or changes in gem cells passed on to the next generation
yeah,
because they couldn't follow during the embryogenesis, right?
yeah.
But they see they are coming back in offspring. So it's a huge or big question mark how this information inherited. That probably future research will answer that question. But we do have, as you mentioned, Dr. Madsen, from the human cohort from Second World War, the Dutch hunger winter, or the hunger winter, right? Where the mothers had issue with the food because the German
Yeah.
army blocked food
Yeah.
supply to the Dutch Netherlands, right? I guess.
Yeah.
And then some of the mothers are during early pregnancy. Some of them are late pregnancy. Then there were few studies looking at the phenotypes or the children, and they found they are suffering from metabolic disease, mental disorders, and etc. So that there's a clear
Yeah. So
um,
you can, I guess the point there is that it can go the other way. So I mentioned this challenge
yeah,
recovery. So in this case, it's challenge sustained, no recovery period,
no recovery period. Yeah,
right?
yeah,
So you
yeah.
got to have these. Okay.
Yeah, but yeah, that's but intermittent fasting, it's based on our published data. At least female offspring are doing much better when you challenge
Yeah.
them with the high glucose, which is the diabetic test, right?
Yeah.
And these animals are under high fat, so they develop diabetic. So we tested offspring from intermittent fasted parents are protected compared to offspring from ad libitum. So that's the positive data. And if that can translate to the brain diseases, that's the question we will try to answer in the years to come. Yeah.
Okay.
Yeah.
Okay, Gary. Thanks for taking the time again.
Thank you. Thank you so much, Dr. Madsen.
All right. Bye.
Bye.